Okay, so here's the How Science Works question on the speedy crab. So, let's get into it. Crabs are invertebrates that live on the seashore. Scientists investigated the digestive enzymes produced by 3 species of crab. They measured the activity of 3 enzymes: endopeptidase, cellulase, and luminerase. So endopeptidases, clue here, peptide, they are going to digest proteins. Cellulase is going to digest cellulose. Cellulose found in the cell wall of all plants and algae. Luminerase is digesting a substance that's only in seaweeds. So here we go. Red crab, loads of cellulase, not so much of the other 2. Shore crab, bit of all 3. The speedy crab, loads of endopeptidase, very little of the other 2. Then we have a food web, seagrass, a plant, seaweeds, the only things that are going to contain the substance that luminerase digests, and then algae. Mollusks, barnacles, and our crab species X, Y, Z. Our first question. Can we identify the species of crab Y and Z and give reasons for our answers? So here we go. So Y is eating animals and seaweeds. So it's eating seaweeds, and it's the only crab that's eating seaweeds. So I'm going to say I'd expect it to be the only crab with a high concentration of luminerase. So luminerase, well, there's only one crab with a lot of luminerase, which is shore crab. So why is the shore crab? Our reasons: it's the only crab that eats seaweeds, and therefore there is only one crab that eats seaweeds, crab species Y, and the shore crab is the only crab with a high concentration of luminerase. Species Y also eats some animals, so we would expect some protein digesting endopeptidase, and the shore crab also has some protein digesting endopeptidase. So 3 things we can say there. Species Z. So species Z only eats animals, barnacles. So we wouldn't expect much cellulase, we wouldn't expect much luminerase, but we would expect a lot of endopeptidase. So it's going to be the speedy crab. So what's the reasons? The reasons then, it's species Z only eats animals. It only eats barnacles, which contain a high concentration of protein. And species Z, the speedy crab is the crab with a high concentration of endopeptidase. So let's get on to this one. The activity of the endopeptidase from one species of crab was measured. A sample of the enzyme was added to a solution of a colourless substance called BAPNA. A yellow compound is produced when BAPNA is broken down by the enzyme. Endopeptidase plus BAPNA, which is colourless, turns into a yellow compound. Describe how you'd carry out an investigation to find the optimum temperature for the activity of the endopeptidase. So let's think what we're going to do, and then let's decide on the most important information to get 3 marks. Here's what we'd really do. We'd have a range of water baths to maintain constant temperatures at a range of temperatures. So here's my water baths at a range of temperatures. Then at each temperature, I'd mix some enzyme and some BAPNA, and I would time how long it takes for it to become yellow. But they're saying an investigation, and to make sure I do it and get results which I can compare, in each case I need to use the same amount of endopeptidase and the same amount of BAPNA. So, I need to explain that I would obtain a range of different temperatures using water baths. That in each water bath, I would mix some endopeptidase and some BAPNA, and would time how long it takes to turn colour, to turn yellow in colour. I would maybe, that's 2 points, I would maybe define my end point. So we've got the range of temperatures, we've got the time reaction to yellow. I might say I would use a colour standard, or I'd use a yellow card, then I would time how long it takes to match the colour on the card. And the last thing I would say is, I would say at each temperature I would use the same volume of enzyme solution and BAPNA. And that should do it. Thank you very much.